Welcome to today's ERSDR Kitchen episode with the theme Freshly Baked, which are dedicated to the latest high-impact findings in investigative dermatology. So my name is Marianne, and I'm chairing this session together with my colleague Chris. And we are very delighted to have two excellent speakers to present today, Laura and Paula. And after the presentations, we will have a Q&A session. So please remember to put your questions or submit your questions during the talks. And of note, this session is being recorded and it will be available on the ESDR YouTube channel. So with that, I will start by introducing the first speaker, which is Laure Guenin-Macé, and she is an associate research professor at the Pasteur Institute in Paris, where she is specializing in host microbe interaction in the context of infectious and chronic inflammatory skin diseases. And Laure, we are very much looking forward to hear your presentation, so please go ahead.
Yeah. So I will share my screen and sorry. Is it good? Can you see my slides? You see my slides?
Yes, it's very clear.
Okay. Yeah. Okay. Sorry. So thank you for this introduction and for this opportunity to present this work that is focused on the role of dysbiosis in hidradenitis suppurativa disease, a work that was featured recently on the cover of EMBO Molecular Medicine. So HS is a multifactorial chronic inflammatory disease of the pilosebaceous unit that occur mostly in skin fold area and manifest by the formation of painful nodules, abscesses, and in severe case, this will evolve into ulcers with formation of tunnels with purulent discharge. The disease is complex and involves factors associated to lifestyle, such as smoking, high BMI, but also hormone and genetic factors. It has a high impact on the quality of life of patients because of the anxiety and the disability and as well as the pain. And currently, there is no treatment to cure the disease, but mostly symptomatic treatments consisting in antibiotic combined to immunomodulators and surgery. The pathophysiology is complex and not fully understood. And the current model integrates etiological factors occurring in prone skin area. And the disease is believed to start by hyperkeratosis and follicular occlusion that will favor the expansion of an anaerobic flora. And this expansion will lead ultimately to the rupture of the hair follicle, releasing high quantity of damage and pathogen-associated molecular pattern. This will trigger massive immune cell infiltration, including neutrophil, macrophages, and adaptive cells, including Th1, 17, and B cells. So as the disease progresses, the increase of the expansion of the dysbiosis of the bacteria, together with the uncontrolled immune response, will lead to tissue destruction and scarring. So although the disease is not an infectious disease, the skin dysbiosis is a hallmark of the disease that has been well documented. It is believed that the dysbiosis play a role in maintaining and sustaining this chronic inflammation, especially through the persistence of biofilm in epithelialized tunnels. So as the disease progresses, the skin microbiota will evolve from a community mostly occupied by common source toward community with a lot of anaerobe species. And the expansion of the anaerobe species is correlated with the severity of the lesion. It has been documented that there is a high increase of Prevotella, Porphyromonas, and Fusobacterium associated with the severity of the lesion. And to date, the antibiotics remain the first-line pharmacological treatment for the patient. So while this dysbiosis has been well documented, its contribution to the pathophysiology is not understood. And very few limited number of studies have investigated this. In a previous work, we had demonstrated that the dysbiosis is associated with an impaired tryptophan metabolism at the host microbial interface with a defective activation of AHR signaling, providing a link between the dysbiosis and the uncontrolled inflammation. However, what is the role of individual bacteria species and how they interact with the host is unexplored. And we believe that some specific anaerobic bacteria may drive this chronic inflammation, and that patient could mount a humoral response against skin microbiota. And this hypothesis was supported by the fact that it was reported an increased expression of immunoglobulin and plasma cell signature in the skin of HS patient, together with the presence of tertiary lymphoid structure within lesion in a severe patient. So to investigate this, we had access to bacterial clinical isolate from patient together with serum sample, spanning all the 3 severity stages from HS patient, from mild to severe. So we had access to a collection of bacterial clinical isolate that were incubated together with a serum, and we measured the species-specific humoral immune response by flow cytometry. And this work was carried out by Vivian, a postdoc in our lab. So here on the left are shown the relative level of circulating IgA and IgG reacting toward these bacterial species in serum from HS patient to compare to LC control in the 3 different severity group. And you can see that the sera from patient react with several bacterial species, and this is particularly true for the sera coming from a severe patient with high reactivity against mostly Porphyromonas renonis and Prevotella bivia. And these bacterial species are increased in severe lesion together with the concomitant decrease of the commensal Cutibacterium acnes. So focusing on these 2 bacterial species, Porphyromonas renonis and Prevotella bivia, we analyzed the type of Ig that target these 2 bacterial species in a severe patient. And we showed that there is a classical Ig response allowing complement activation and opsonization, together with increase of IgA, and which was striking was this increase of secretory IgA indicating a rupture of the barrier and supporting the infection-driven hypothesis. Next, we use a bacterial specific mRNA probe on LC skin and skin from different patients with severe form of the disease. And we show that only Porphyromonas renonis and not Prevotella bivia was present in the skin. And the bacteria was present not only in the epidermis, but deeply in the skin at the dermis level and distantly from the lesion, supporting an active invasion of the bacteria. Together with the high humoral response against this bacteria and the invasion of the tissue, we showed that the skin invasion coincided with the recruitment of CD138 positive IgA producing plasma cells. So altogether, these data in patients support the fact that Porphyromonas renonis is a skin invader able to trigger local and systemic humoral response in a HS patient. Next, we moved in vitro to understand the invasive and pro-inflammatory capacities of the bacteria. Using primary keratinocyte, we showed that the bacteria was able to enter and persist into keratinocytes and to trigger a pro-inflammatory response. And in comparison, Prevotella bivia only triggered a mild inflammatory response. Next, we use a LC skin explant and deposition of the bacteria on the top of the explant. We showed that Porphyromonas renonis was able to actively invade this LC skin and again to trigger a pro-inflammatory response with high production of IL-6 and CCL20, showing that the bacteria has the capacity to invade an LC skin. And finally, we moved to the in vivo model using the model of topical association of bacteria developed at the NIH in the group of Yasmin Belkaid, and this work was led by Inta Krebenica. The bacteria was associated on the skin of immunocompetent mice for 4 consecutive days, and sera and skin were collected 14 and 60 days post-association to check infection, recruitment of plasma cells, and antibody production. So with this model, we could not detect the presence of the bacteria within the skin, and there was no development of abscess or ulcers. However, we find that there was development of humoral response that developed over time, it peaking at day 60, with high elevated level of serum IgG1, IgG2b, 2c, as well as IgA and secretory IgA, which is similar to what was observed in HS patient. Using whole mount imaging, we showed that there was an accumulation in the skin of CD138 positive plasma cells, and this was specific to the association with Porphyromonas venenis, as it was not observed with Prevotella bivia. And this accumulation of plasma cells within the skin was observed till day 60. And this was confirmed as well by flow cytometry. So again, this phenotype of plasma cell accumulation strikingly mimicked what we observed in human skin from HS patient. So as a conclusion, we shed light on the long debated role of the dysbiosis, on the contribution of the dysbiosis to the pathogenesis, showing that patient mount specific antibody response against skin colonizer, and especially against Porphyromonas venenis, which is selectively enriched in severe disease. Porphyromonas venenis is normally a rare inhabitant of the skin microbiota, but we show that it is able to invade and activate human keratinocytes, even healthy skin, and trigger local humoral immunity. And we propose that this combination create a self-amplifying inflammatory loop that is chronically sustained by bacteria present in the lesion. And our results support that Porphyromonas venenis could be a driver of severe form of HS disease with direct implication for prevention and treatment. And with this, I will thank the people who participate to this work, especially the group led by Caroline de Mangel at the Institut Pasteur with Viviane Bobin, all our collaborators, and the team of Yasmin Belkaid at the NIH, especially Inta and Jeremy. And finally, I would like to thank my new team, Meta-Organism, led by Yasmin Belkaid in Pasteur, where I will pursue this work. And with this, I'm done.
Thank you so much, Laura. That was an excellent, clear talk. Chris, you will take it from here?
Can we ask questions?
No, in the end.
In the end. Okay.
Yes.
Very well. I'll leave it for the end. Thank you for this marvelous presentation, and we bring the words to the next speaker. Please welcome me, Dr. Paula Restrepo, a computational biologist who just completed her PhD in genetics and genomics at Icahn School of Medicine at Mount Sinai, New York. As now a postdoctoral fellow there, her doctoral work built a first-of-its-kind single-cell spatial atlas of the human skin, published in Nature Genetics recently, and her postdoctoral research focused on multimodal foundational models predicting immunotherapy response across cancers. We are all ears. I'm very curious to see your presentation. The floor is yours.
All right. Can you see? Thank you so much for the beautiful introduction and for the invitation. Give me 1 second. Can you see my slides?
Yes, very clear.
Okay, great. So good morning, actually, from New York City. I'm looking forward to presenting a recent Nature Genetics paper, as well as, which is also basically some of my thesis work. So to kind of get started, to introduce a little bit, the skin is this super complex cellular ecosystem, as we all know. that varies in its cell composition and its spatial morphology, not just within the tissue, but also across the body on a macroanatomic level. And so this intricate patterning that varies spatially across the body, this is reflected by the fact that certain anatomic sites are more predisposed to certain skin diseases than others. So for example, atopic dermatitis tends to present on flexural areas, whereas psoriasis tends to present on extensor areas. But we noticed we kind of reasoned that in order to understand the site-specific disease predilection, we first have to understand what is the normal baseline homeostatic heterogeneity across the body. And we noticed a major gap in terms of understanding the tissue intrinsic molecular features that underlie the spatial patterning of the skin microenvironment. So in this study, our major goal is to understand how do cell types and communication networks change across the spatial patterning of the skin. And so in order to understand the skin microenvironment in homeostasis, both within the tissue and across anatomic sites, I sought to construct a multimodal atlas-style resource with spatial profiling with a novel cohort of 114 normal skin samples across 22 individuals. So I used spatial transcriptomics with MERFISH, that's a subcellular resolution spatial transcriptomics assay. And then I also leveraged the wealth of public data sets within the literature and integrated my data with single cell and spot-based spatial transcriptomics data sets. And importantly, we're leveraging a matched sampling strategy using autopsy donors where we were able to sample multiple, up to 12 different anatomic sites from the same individual donor. And so today, I hope to walk you through not just how I built this atlas, with the overall goal of understanding the molecular diversity of normal human skin, but also how I've used it to identify cell types and cell subpopulations, identify neighborhoods, and compare the diversity of the cellular populations, both across anatomic sites and within the tissue, and also looking at how that organization changes in disease when things go wrong. So I mentioned I had that novel cohort with the matched sampling strategy. And what did we do with those samples? We did MERFISH. So MERFISH, or multiplex aerorobust fluorescence in situ hybridization, is an in situ spatial transcriptomics platform that is based off of single molecule FISH, where each gene in a custom panel gets a unique binary barcode. And so using sequential rounds of imaging, these barcodes are identified and they're decoded, and this lets you detect up to 1,000 genes in your tissue. In our case, we designed a custom 500 gene panel composed of roughly equal parts cell type markers and relevant ligand receptor pairs. And so using MERFISH, we were able to detect all the transcripts, all the RNA transcripts across the whole tissue slide for all 114 samples, as shown by this close-up of a hair follicle where each point is an individual transcript. Using cell segmentation algorithms, we can draw boundaries around each cell, and then we can assign transcripts to cells. So we ultimately end up with a cell by gene matrix, just like any other single-cell RNA-seq data set, where we can analyze this with standard methods like unsupervised clustering and UMAP visualization, but we also have the spatial coordinates for where each of these cells localized within the tissue. Okay. So using this approach, I have profiled over 1.2 million cells from all 114 samples, as shown by this global integrated UMAP, not only identifying major cell types within the skin, but also, as I mentioned, where each of these cells is localized within the tissue. And so here I'm showing you one of our donors where we have all 12 anatomic sites profiled. And each point here is a cell colored by the cell type that was identified by the unsupervised clustering. And while in the interest of time, I'm not going to get into it, we were able to do subclustering across immune, stromal, and epithelial compartments and provide a really detailed catalog of cell subpopulations across the skin with kind of deep phenotyping that's comparable to single-cell RNA-seq, which is the gold standard, as well as very precise localization within the tissue. So now that I have my cell types, I wanted to do some differential abundance to basically look at how the abundance of each cell type changes across the anatomic sites. And so in order to deal with covariates and really take advantage of this donor-matched cohort, I used a method called Crumbler, which allows you to model count and proportional data with precision-weighted mixed linear models. And so this is going to allow us to take care of, deal with those covariates and also take advantage of the fact that we have matched samples. And so this is going to be a busy plot, but I'm going to walk you through it. So what I've done here is some one versus all comparisons for each cell type, where I ask if each cell type is more or less abundant in a given anatomic site versus the average of the rest. And the variance partition bar plot on the bottom here tells us how much compositional variance of the cell type across the anatomic sites is driven by each term in my models. And as you can see with this red here, a lot of this compositional variance is being driven by anatomic site. If we look at the top at this hierarchical clustering, we can see that there's this interesting gradient with clusters of diverse cell types that are very specialized. Like, for example, this reticular fibroblast 2 population that was ANGPTL7 positive that we found that was localizing very specifically to the sole. We also can see how we see an enrichment of immune populations in the antecubital fossa. There's also some stable cell types like the eccrine glands that don't appear to vary so much across anatomic sites. And we can also look at specialization in terms of our rows. So for example, we see that the hair-bearing areas like the face, the scalp, the face and the scalp appear to be very specialized in terms of certain populations localizing specifically to them, as well as some anatomic sites that are maybe more generalized, like the butt, the belly, and the back, where there's not really so many populations that are specifically localizing to those anatomic sites. And so in summary, we've identified these areas of diverse and stable cell types, as well as populations that are more generalized and specialized across the body. And so I took this a step further to look at how the cells are organized in the skin. And so just like how New York City is made up of the boroughs and the neighborhoods and all the great people that live here, we can also look at how cells organize into neighborhoods, which in turn make up the tissues of the skin. And so cellular neighborhoods are this very recently emerging concept in spatial biology, where instead of looking at these really broad anatomical compartments or going all the way down, zooming all the way into each cell's immediate neighbors, these cellular neighborhoods are defined recurring structures within the tissue with characteristic resonant cell types and cell interactions. And we can identify these neighborhoods by combining both molecular data and spatial coordinates to build upon traditional histology, allowing us to not only reproduce known tissue domains, like say the hair follicle, but also to identify new ones. And so to this end, I used a published method called Cell Charter to identify neighborhoods, and this is going to allow me to directly quantify the spatial heterogeneity of which cell types reside in each neighborhood and how they're communicating with each other in the skin. So I found 10 multicellular neighborhoods in my data set, as shown by this example representative tissue slice where each cell is colored by the neighborhood in which it resides. And each of these neighborhoods has a distinct localization pattern within the tissue, as shown by these spatial highlights across my 10 neighborhoods. They are also multicellular communities, as you can see by their demographic breakdown in this heat map of cell type proportions. And using this approach, we were able to find these very well-established adnexal structures like the eccrine glands, as well as some potentially novel and very interesting tissue niches like this perivascular neighborhood, which was surrounding these small blood vessels kind of around the vasculature and was particularly enriched in immune populations. And so now I'm going to switch gears a little bit and dive into one of these neighborhoods. And these neighborhoods that are these diverse communities have these distinct communication patterns, which we can detect because we carefully designed our 500 gene panel to both deconvolve cell types and identify signaling interactions. So this perivascular neighborhood had this specific localization of immune cells. And if we look at which pathways are activated in terms of signaling within this neighborhood, we can see several immune signaling pathways like CXCL, CCL, and TNF. I then wanted to know which cell types were most responsible for driving signaling via cell chat within this niche. And here we can see that T cells appear to be our biggest receivers of interactions, and these CCL19 positive perivascular fibroblasts were the biggest senders. And so if I directly visualize this and look at specific interactions, I can see, for example, there's this TNF signaling from T cells to perivascular fibroblasts that was especially compartmentalized to this niche. And this is important because it hints at the skin-associated lymphoid tissue, which is this concept that was introduced in the late 70s by J.W. Strelin that proposes that the skin can act like an immune organ. And so based off of the evidence that we see, we are kind of proposing that this perivascular 1 neighborhood is a direct correlate of homeostatic salt, and that it's likely to host these homeostatic immune surveillance. And so this potential made it, this kind of observation made this niche very kind of an interesting candidate for functional validation. And we wanted to know if this spatial compartmentalization of TNF signaling held up in functional validation. And this was interesting because in previous studies of inflammation, there's been evidence that TNF signaling from T cells can cause fibroblasts to upregulate CCL19. So we asked, can TNF increase CCL19 expression, and is this spatially restricted? So using skin, a postdoc in the lab took intact human skin as explants and cultured it. And with the fresh explants, she saw that we see these CCL19 positive perivascular fibroblasts in this perivascular space, specifically using RNAscope. But if she cultures it for 48 hours, then the CCL19 expression is not maintained. But if she cultures it in the presence of TNF, we see that the CCL19 is maintained. And so this really suggests that there's a spatially compartmentalized TNF signaling between T cells and fibroblasts that's maintaining this identity, this kind of interesting homeostatic feedback loop within this surveillance niche. And so lastly, very briefly, we wanted to look, now that we have these normal neighborhoods, we wanted to know what happens to them in disease. And so we looked at publicly available spot-based spatial data across 5 different skin diseases, as well as normal skin, as shown by this integrated UMAP of all 142,000 spots. And by clustering these, our rationale was that the spot clusters were able to capture the neighborhoods that we could map back to our MERFISH neighborhoods that are present in normal skin. And so then using the same differential abundance approach that I used for cell types, I looked at whether the neighborhoods are expanded or contracted in disease versus normal skin. And interestingly, I found that this perivascular 1 neighborhood is expanded in 4 out of the 5 skin diseases that I looked at. And you can also see this by these kind of spatial highlight plots, kind of visualizing it in some representative tissue slices. We also looked at the chemokine expression, and we found that the perivascular neighborhood also appears to go transcriptional remodeling in terms of chemokines, interleukins, and extracellular matrix remodeling genes. And this is interesting and important because it implies that this spatial niche is potentially targetable for the development of therapeutics. So in conclusion, we basically built this refined blueprint for normal human skin, creating this large-scale single-cell resolution spatial atlas as a resource for the skin biology community. And we've identified these neighborhoods that are effectively building blocks for normal skin. We've identified this perivascular neighborhood that's hosting homeostatic immune surveillance, and we've also looked at how that goes wrong in disease. And importantly, we've made the resource browsable with a web portal, and we've made the data available, the raw data even available for the community, so it can be used even for the development of things for kind of like outside of skin biology for, things like algorithm development. And with that, these are my acknowledgements. I'm sorry, I know I went a bit over. And I just especially want to acknowledge my thesis advisor, Dr. Andrew G. And with that, thank you.
Thank you so much, Paula. That was an excellent presentation. So we will just have time for a few questions, and I'll just go to the Q&A. And Laura, the first one goes for you. So thanks for the nice presentation, Laura. Does fungi have any role to play in HS beyond the bacteria?
So all the studies done on the dysbiosis, I'm not aware of any study reporting an increase of fungi. It seems to be mostly an expansion of anaerobic bacteria, but I don't know if this has been studied very deeply.
But you used like metagenomic, right? So is it possible to have a look in the data, or was the sample prepared only for the bacteria?
So the data come from our collaborator, and they focused really on the bacteria. I don't know if we can access to this level of information. I could check anyway. Yeah.
Okay. So there's another question from Michelle Simon. Do you know how keratinocytes are activated by a PU or through TLR binding? Question mark.
Say it again, sorry.
So how the keratinocytes are activated by the bacteria?
Oh, sorry. Yeah. So this is something we are currently investigating, and we show that the keratinocytes are activated by both contact with the bacteria and diffusible factors, and this is very compatible with the TLR activation. But we need to we will investigate precisely what are the mechanism of activation. But anyway, it fits perfectly with TLR4, TLR2 activation, the kind of signature we get from the keratinocyte.
Okay, Chris, are there any questions in the chat?
Yes, also two questions for Laura. Laura, in your mouse model, did you investigate the commensal flora at the same time as well?
Not this time, but again, this is ongoing work, and we'll pursue this work of topical association and check how the presence of the bacteria impact the commensal microbiota, and whether it needs other HS-associated bacteria to really colonize the skin.
And there was another question to Paula. Amazing presentation. How did you match back the neighborhood found in your MERFISH data to the online ST spot data?
Yeah, that's a great question. So we took two basically major approaches. Our rationale was that in each spot, since you have like up to about 10 cells, each spot is kind of like a mini neighborhood already within the spot-based data. So we used expression, the kind of cluster markers, and we mapped that based off of similarity. We also used the spatial localization patterns, just kind of manually annotating it. And then we also used cell to location to deconvolve those spots to look at which cell types would we're making them up, and we compared based off of the cell composition. And so those are the major approaches that we used, and we basically took an annotation-based approach to kind of map and harmonize.
Wonderful.
Okay. Thank you.
No further questions here in the chat. I think we are a little bit over time. Maybe we can round up. And we'll pass it on to Lea.
Yes. I would like to thank the two speakers for amazing presentations, and also a huge thanks to all of you who joined us today. So this is just a reminder that all these webinars are open access and will be available on the ESDR Kitchen YouTube channel. And the next ESDR Kitchen episode will be in August 26, where Professor Hans Mandal will talk about some of his work in glycomics. And then also I would just point your attention to the ESDR Skin Chronicles, which is an initiative where we try to highlight publications that have made significant contributions to translational dermatology. And the authors here, they are all volunteered, and they have, write these pieces, and they have chosen papers that they want to share with the communities. And if you're interested in contributing to this initiative, you are more than welcome to reach out to me. And then finally, I just want to point out that we have the ESDR annual meeting in Heidelberg, and I hope to see many of you there. So the late break-in abstract is up for July 12th, and early fees end 8th of July. So if you didn't sign up already, please do so. And if you are joining the ESDR meeting, please come on Wednesday to the Young ESDR Symposium. We have a great lineup of speakers, and it's a very good opportunity to network you with your research fellow. So I hope to see you there. So with that, I will close the session, and have a good day all. Bye.